All right, everyone. This is Dr. Granier, and this is Chapter 17, which is our special senses chapter for our nervous system. So we have 5 special senses, and this information goes somewhat with Chapter 15 in our general senses. So the big thing that you should take away from Chapter 15 and 17 is the general senses are going to be found throughout the body. They're mechanoreceptors, they're chemical receptors. They are going to be carried through ascending tracts towards the primary sensory cortex of the parietal lobe, the postcentral gyrus area. And what's going to go on in the special senses is they are located in some unique anatomical spot. They have a very unique receptor system, and in many cases are being carried by one of the cranial nerves, and they don't have to go to the primary sensory cortex. They may go to, like in the case of vision, they're going to be going to a complete lobe that's almost dedicated to vision, the occipital lobe. Temporal lobe is going to play a role in this with some of the other senses, especially with taste, what am I trying to think? Equilibrium and balance and hearing. So and the cranial nerves are going to come into play. And so this is one of the reasons why I make you learn your 12 cranial nerves, because we utilize them with the special senses. Okay? So olfactory is smell, gustation is taste, vision. Equilibrium, you have dynamic and static equilibrium. So dynamic equilibrium is the movement side, that's the motion sickness component. And then the static equilibrium is basically with knowing you're up and down, the horizon. And so people with vertigo, they sometimes, they don't know that they're standing still, and they kind of feel nauseated because of that. And then the auditory hearing component. Okay. So that's what we're going to do. There's a lot of slides, so bear with me. All right, for the olfactory, we have to look at, this is the nose. These are chemoreceptors that are going to interact with airborne particles by trapping them in a mucus. And then with that mucus interacting with a chemical receptor, that's going to lead to changes in the neurons that are extending down from the olfactory bulb and part of the olfactory nerve. And then those changes will lead to, of course, an action potential that would then travel along cranial nerve 1, the olfactory nerve, through the olfactory bulb, through the olfactory tract, and then have an influence on understanding smell. All right, and we have lots and lots and lots of smells, and they are going to end up somewhat tied into the limbic system, because as we know, if you smell like homemade cookies, that can make you feel comforting and happy. And we actually, use our smell to taste, so with eating, to help pleasure with food. All right, so the olfactory glands are secretions that are coat surfaces of these olfactory organs. The receptors are very highly modified neurons that allow detection of chemical stimuli, and they are going to bind to these chemicals that are in the air that get trapped into the mucus secreted by the glands, and then that interaction of chemical and receptor on the neuron will lead to changes that allow potential action potentials to be sent. Okay? So axons that leave the olfactory epithelium collect into 20 or more bundles, so little fascicles. They penetrate through the cribriform plate of the ethmoid bone, and they are going to then reach that olfactory bulb, that kind of enlargement of the olfactory nerve, and that's where they're going to have their first synapse. So the first order neuron then is that neuron tied to the receptor that goes to the olfactory bulb. And think of the olfactory bulb kind of like a ganglia. It's in place where a synapse is occurring, right? And then the second order neuron is going to leave that olfactory bulb and is going to travel to the olfactory cortex, so an outer region of the brain that's going to be tied to olfactory and smell. The hypothalamus, and again, because there's ties of smell and emotion, some of the areas of the limbic system, which sits right on top of the corpus callosum, is going to get information from these neurons. And the arriving information is going to reach the information centers without synapsing in the thalamus. So when we looked at our general senses, they almost always went through the thalamus. That's like a big gatekeeper point. So this is one of the places where the thalamus is bypassed. So it's not a gatekeeper for smell. Okay? When we look at taste, it's known as gustation. It provides information on foods and liquids that we are consuming. So we have taste receptors, which are known as gustatory receptors, and they're distributed on the tongue in portions of your actual pharynx and larynx, so the back of the throat and even towards the, going into that area, the esophagus and the epiglottis region. They are clustered together, these receptors, into little areas known as taste buds. And so when we look at it, we have 3 types of linguinal papillae. We have the filiform papillae, which is down here on the bottom. Okay, and the filiform papillae, their purpose is providing friction, and they really don't contain taste buds, but they're helping with some of the mechanical processes of breaking down food. The fungiform papillae are going to contain 5 taste buds each. And then our circumvallate papillae will be a large portion of taste buds, so you can kind of see them in that little region there. Okay. The taste buds contain these basal cells. Then they contain gustatory cells, which are going to be specialized cells that have taste hairs that are going to extend into the lumen of the cavity. And they are going to be what is survivable for about 10 days and then are constantly replaced. So part of the reason why your taste changes over your lifetime is because these cells are constantly changing. The taste is going to be tied to cranial nerves, and because they're located both on the tongue, the pharynx, and the larynx, there's a few cranial nerves that actually carry information for taste to the central nervous system. The taste is going to head on the thalamus, and so therefore it is going to stop at the gatekeeping site in the diencephalon. And then most of your taste actually does head to the primary sensory cortex of the postcentral gyrus in the parietal lobe. Okay. Oh, that went too fast. All right, so what cranial nerves are involved? So the facial nerve, which is cranial nerve VII, so that's 7, it innervates all of the taste buds located on the anterior 2/3 of your tongue. So it's going to be the tongue proper for the most part. So from the tip of your tongue to the line of the circumvallate papillae, that's where your cranial facial nerve is going to be located. The glossopharyngeal nerve, so pharyngeal's in there, so it's going to nervate more of the back end of the tongue. The vagus nerve, so cranial nerve 10, actually innervates taste buds scattered around your epiglottis. And so plays a role in a little bit of that swallowing and making sure, you know, things are not going into the lungs. And then your trigeminal nerve, which is the big nerve with the 3 clusters, cranial nerve number 5, that's going to play a lot of, it's a mixed nerve and plays a lot with sensory and motor control of a lot of the features of the face and tongue and the lips. It's going to give information for food that's hot or peppery. So some of those unique, not necessarily sweet, sour, salty, but some of the umami, some of the special like wasabi and the hot peppers and things like that. It's going to play a role in that information in the tongue. Okay? So questions that I would ask, I could ask you on lab practical or lecture, which cranial nerve innervates the top third of, top 2/3 of the tongue, which one's the back third, which one's around the esophagus, and you would be able to recall by name or number. All right, so you have 4 primary taste sensations: sweet, salty, bitter, and sour. And then you will have 2 additional types of taste receptors for water, which are more towards the pharynx and the very posterior parts of the tongue. And then umami, which is, comes from a little bit of like a Japanese sounding, which is related to that seasoning, okay? So when we look at it, again, our circumvallate papillae, so you do most of your tasting towards the middle and back third of the tongue. And the very front is mostly about texture and about feeling and mechanoreceptors with those filiform papillae. When we look at the histology, we see that taste buds, those gustatory cells, are going to be the hair cells that are going to try to express into the spaces, so the interactions of the food. And then the basal cells are going to be located towards the inner regions and tied to the neurons themselves. Okay. So again, your anami is going to be for beef, chicken broth, Parmesan cheese, some of the receptors specific to amino acids, peptides, and nucleotides. So things that related to that have nitrogenous components. And then water is, of course, going to be with the pharynx. And then this is sometimes why you might feel like water has a taste or a feel to it. Why does it do that? Okay. So where's taste going? So the point of taste, how does it work, is there's release of neurotransmitters by our receptor cells, our gustatory cells. Those gustatory cells are going to work on the dendrites of sensory afferent neurons that are wrapped by receptor membrane, and it's going to generate can generate action potentials on those sensory neurons that then carry that information to the central nervous system. Okay? So that's really the big overview of taste and smell. So now we're going to go into the eye. All right, the eye. The eye, of course, is going to be where we're looking at vision, and the point of vision is getting the light to end up at one specific area where we maximize our ability to focus, interact, and have electrical currents carried by cells that enhance what we're being stimulated with light. But in order to get the light to that right spot, we have some accessory features that help protect, lubricate, and support the system of the light fixing. So the eyelids, the real name for them is the palpebrae, and so that is one of the big features that helps protect and helps it lubricate and continue to wash the cornea with liquid, so therefore you prevent potential drying out of the eyes. Right? When we look at the eyelids, there's superficial epithelium. So there's going to be some epithelial cells self-touching each other, providing the actual covering. And we look at the lacrimal corniculi as a massive small tissue, and it contains some glands that produce secretions. And the secretions, again, are going to have a little bit of enzymes to help keep things from trying to, infect and take advantage of the eye as an entry point, as well as secretions that are going to keep things lubricated. All right, and every now and then you wake up and you feel like you have crud in your eyes, and sometimes that lacrimal corniculi is what is contributing to that crud development after a good night's sleep. The conjunctiva is epithelium that covers the inner surfaces of the eyelids. So when you have conjunctivitis, this is inflamed, and therefore you have pain on the eyelid because this is every time you're moving and manipulating and mechanoreceptors of the eyelid closing on the eye, you're manipulating this conjunctiva, and it's causing action potentials and discomfort. All right? And then you have a lacrimal apparatus, which is going to be involved in the watery secretions. Okay? So the lacrimal apparatus distributes, and removes tears. And so a fornix is a pocket where we have the palatal conjunctiva joins the ocular conjunctiva, and it's just this little space. And then you have the tear gland, which is known as the lacrimal gland. And again, you're going to secrete an enzyme known as lysosome, which is an antibacterial enzyme that tries to prevent bacteria from trying to get into the layers of the eye, live in the eye, and cause infection of the eye, and then the bacteria trying to get into your bloodstream. When we look at the tears, they collect in an area known as the lacrimal lake, and that lacrimal lake is a series of little passageways. So you have the lacrimal punctua, the lacrimal canuliculi, so two little kind of like connecting ditches that collect into a sac, the lacrimal sac, and then you have the nasolacrimal duct, which is an entry point of how tears end up in your nasal cavity. All right. So when we look at it, here is the lacrimal punctum right here, right? And you have the lacrimal carniculae and the medial, and then you have your little caniculi, the sac, the duct, and then an opening into that nasal cavity. So this is why sometimes excess water might end up dripping out your nose. Okay. The gland is more of a lateral thing. So when we look at our eyeballs, one of the ways we can tell medial versus lateral is we know the gland is going to be located on the lateral surface of the eye. Okay. Let's see. We have the ocular conjunctiva, which is going to be as close to the eye, and then we have the other conjunctiva. Let's see. They show it. No, I don't see it. Okay. So those are just some of those features, right? Now, actually getting into the eyeball, there are 3 layers to the eyeball. You have an outer layer, an inner layer, and then the very deep inner layer. All right, the outer layer, fibrous, should make you think of collagen-rich connective tissue that's strong, that helps give shape and function. So the fibrous layer is going to be composed of the cornea, which is going to be the clear area where light passes through, and then a white area known as the sclera. The intermediate vascular layer, which is known as the choroid plexus, is going to be heavy with pigmentation, because that's going to help prevent the scattering effect of light. So it helps absorb scattering rays of light. And so there's a pigmentation, so there's going to be lots of melanin produced by melanocytes. And then you're going to have blood vessels coming into here because, of course, there are cells in this area, an inner area and outer area, that need fuel and oxygen. And then there's going to be some other neural components that are going to talk to and control other areas of the eye. The innermost layer is going to be your retina, okay? And the retina is going to be where we're going to see neurons and everything else related to control of information that's generated from the rods and cone cells, okay? So 3 layers. The outermost is the fibrous, so that's the cornea and the sclera. The middle layer is the choroid plexus, and it's a vascular layer with some pigmentation. And then the deep inner layer is going to be the retina, which is going to contain the photoreceptors, the physical receptor cells that get triggered by light, and the neurons that connect that information to the optic nerve. Now, the eyeball is hollow. It's filled with fluid, and it's divided into 2 cavities by the lens. Okay, so there is a cavity from the cornea to the lens known as the anterior cavity, and that's going to be where we have aqueous humor. And then from the lens to the retina, that is going to be our posterior cavity, and it's going to be more of a jelly-like substance in there that's going to be our vitreous humor. Okay, so the lens, the iris, the little ligaments that control and manipulate the lens, all of that tends to be part of our vascular or our middle layer. Okay? I don't know why it's going so fast. Okay, so this is just a different picture. So the anterior or the outermost layer, the cornea is clear cells, but then we move into the sclera, which is going to be white. And that white connective tissue is going to be important for all the muscles to attach to. The choroid plexus is going to contain the iris, the ligaments, the lens, and then this melanin-rich layer that's going to have lots of blood flow, lots of blood vessels, lymphatics, that's going to be in between the sclera and the retina. Okay. The retina, which is the innermost layer, is going to be, in many cases, it's going to show up pink. And it's going to be the blood vessels and the entry and exit points for inside, and it's going to be lined with rods and cones and the neurons, the bipolar cells that connect to the rods and cones. Okay. From the lens to the cornea, you have an anterior cavity filled with aqueous humor. And then from the lens to the retina, that big hollow sphere is going to be the posterior chamber, and that's going to be filled with vitreous humor. Okay. So the fibrous layer, the sclera, is known as the white of the eye, and then you have the cornea, which is the clear portion in the front. The corneal limbus is the border between the cornea and the sclera. Okay. And these are going to be, again, the sclera is going to have lots of dense, regular connective tissue. The cornea is going to be very important to protect and help with the inner surface of the eye. And again, you can scratch your cornea in certain LASIK surgeries. This is where they cut and pull the cornea back. So it's a pretty distinctive layer. Okay. For the vascular layer, which is the choroid plexus layer, it's going to be a route for blood vessels and lymphatics that are going to come into and surround the eye. It's going to be where the iris is located, so the colored part of the eye, and so that is going to manipulate the pupil, the the passageway and allows light to enter, either high amounts of light or less amounts of light. This is going to be, because there's a large vascular component, lots of blood vessels, lots of lymphatics, this is going to be where we secrete and reabsorb aqueous humor that's in the anterior chamber. And there's going to be the ligaments that manipulate and control the shape of the lens. That's essential for focusing light on the retinal surface, depending upon if we want to see an object close or see an object far away. The iris is specific to this layer that contains papillary muscles that change the iris, so therefore it makes the pupil large or small. Right? And the papillary muscles, remember, they're going to be tied to sympathetic and parasympathetic outflow, and so they will change depending upon the autonomic system. Ciliary bodies are going to extend posterior to the level of the ora serrata, and they are going to play a role in moving the ciliary muscles and the suspensory ligaments of the lens. So they're going to help make the lens change shape as well. Okay. The choroid is, sometimes this layer is known as the choroid plexus layer. So it's the vascular layer that separates the sclera from the retina. And it is going to be filled with cells that are secreting melatonin, and then the blood vessels and the lymphatics themselves. So the blood vessels here are all about bringing oxygen, glucose, and nutrients to the cells of the sclera and the ciliary muscle cells and the ligaments and the everything, all the cells within the tissues. Okay. Now the innermost layer of the eye is going to be again, another pigmenting layer so we can absorb light, and then it's going to be a little backwards. You're actually going to have the rods and cones anchored next to the pigments, and then you have the bipolar cells, and then you have ganglionic cells that are going to run towards the optic nerve and connect into, the optic nerve leaving the eyeball. So what ends up happening is light actually is going to flow through, past all these nerves and neurons first, and then they're going to either hit the rod or hit the cone cell and then be absorbed, so you don't get scattering and like kind of bounce back by hitting that inner pigment layer, right? Rods and cones are specialized cells that will change some of their proteins within, known as rhodopsin, based upon light or photon energy. And then based upon those changes, that will trigger changes in potassium and changes in ions that will trigger action potentials on our bipolar cells, which then will, like any synapse, have a neuron-neuron synapse with ganglionic cells. Okay? So technically the light actually passes all of the roadway of neurons before it actually hits its receptor. Okay, so this is a cartoon picture, and then over here we see the representative of ganglionic cells. So these are neurons. Here are the bipolar neurons. Here are rods and cone cells, and then here is our pigment layer, and then here is our choroid plexus, which has more pigments beyond that. All right. This kind of shows a bigger picture of, now we have rods and cones located throughout the retina, all right? But if you notice at this one spot, which is known as the optic disc, there are no rods and cone cells. So the optic disc, because it doesn't have rods and cone cells, is technically a blind spot, all right? Now, our rods and cones are distributed throughout the retina, but our cone cells are actually going to be highly concentrated right here in this macula, this little kind of indentation, which is known as the fovea centralis. And the reason why it's central kind of here is this is where our lens, our cornea, everything that's bending and manipulating light, it's trying to harness all of that light to basically sit and activate the rods and cone cells on this specific part of the retina. So this is where you get that 3 to 3 degree window of focus that's best for your eyes, and then all of this is more like peripheral vision. Okay? And so in our periphery, we do have a blind spot, and there's ways to find that blind spot if you close one eye stare at something straight ahead, and then have something in your periphery, and then as you move, there's a little test we can do in lab. Just remind me. Okay. So the inner part of the eye is going to have the rods and cones attached to the pigment layer, and then it's going to have a neuronal component. And the neural component, that's the rods talk to the first neuron, is a bipolar cell. All right, and so these are going to be those cells that specifically are attached to rods and cones and synapse with them. And then they then synapse with ganglionic cells that are going to actually be carrying action potentials through the optic disc and towards the central nervous system of the eye. So the area of entry and exit for blood vessels and all of these neuronal cells is going to be at the optic disc, which is a region just off of the fovea centralis in the macula, and it is an anatomical blind spot for each eyeball. Now, because we use two eyeballs, we compensate and we cover up this blind spot for the most part because the eyeballs kind of see the same things. But there is a way to find the anatomical blind spot when you close one eye and then use a manipulation technique that I can demonstrate and talk you through. Okay? All right, so back to the chambers of the eye. The ciliary bodies in the lens divide the eye into a large back chamber and a small front chamber. That small little anterior chamber is going to be filled with aqueous humor, and you have it subdivided into a little anterior component that's from the cornea to the iris, and then between the cornea, between the iris, the ciliary body, and the lens, there's a little bit of space, but for the most part, that anterior chamber is filled with aqueous humor, and the posterior chamber is more of a jelly, so it's a virtuous humor. Okay. So the aqueous humor is going to be a liquid that circulates between the cornea and the lens. It's going to be basically released by that vascular network in the choroid plexus, that middle layer, and it diffuses through walls of the anterior chamber into a little sinus, and then it can be reabsorbed with lymphatics and by blood supply and taken back up. So we worry about the pressure of the anterior chamber, and we worry about the making of aqueous humor versus the reabsorption of aqueous humor. And so when you start to acquire too much aqueous humor, too much liquid in that anterior chamber, you can have problems with your intraocular pressure. If you have too little, you could have problems of intraocular pressure, but in the opposite way, it's too low. And so the pressure is important because it helps basically provide some separation between the cornea and the lens and helps retain some of the eye shape. That's important because the cornea does a lot of light bending, and then the lens finishes that light bending. When we test intraocular pressure, we can determine glaucoma, if it exists or not, based upon, again, intraocular pressure. And glaucoma is in a condition within the anterior chamber where we're not getting rid, so our intraocular pressure is too high, and so our aqueous humor is being made faster and more than it's being absorbed. So it's a mismatch. Now the back part of the chamber, from the lens to the retina, is going to be known as the large posterior cavity, and it has this virtuous humor. And the virtuous humor is very much a gelatin mass. And what you'll see in the cow eyeball dissection is it is a big, more jelly mass. And it really does help make the eyeball have a spherical shape, and it gives the eyeball and the retina some support and shape. The lens is made up of specialized fibers and cells that have no organelles or nuclei, and therefore they're terminal. So that's why sometimes people have to have lens replacements. They are filled with crystallins which provide clarity and focus power. If you have a cataract, that's a condition in which the lens starts to get cloudy or opaque, and so it's not clear, and so you start to have less light pass through, and so you start to have vision problems. And we see cataracts in our poor little puppy dogs and kitty cats as they age, all right, as well as in people. For the eye, the whole purpose of the eye is to bend light. So it bends or refracts light, and it refracts light through the cornea and then again at the lens. Now, when the lens has problems with light refraction, that can lead into a few problems. If you have problems transitioning from near vision and far vision, typically it's a problem with accommodation. The lens is not able to change shape to really focus up close or focus far away. And as we age, we see there's some accommodation issues, and so that's why we end up with bifocals and reading glasses. Now, an astigmatism is a condition where the light passing through the cornea and the lens is not refracted properly, and it could be because of, anatomically speaking, you have an issue with the shape of the lens and the shape of the cornea and the way they bend light, and or it could be something you develop through pressures placed upon the eye. Right? And so because the refraction, the light isn't bent properly, you get a distortion to the image. Okay? So looking more at the rods and cones on the retina, our cone cells tend to be a little bit more of a robust cell. They have an outer segment which is going to attach to the pigmentation layer, and it contains flattened membranes, plates or discs that contain lots of visual pigments that are capable of responding to all the different colors of the Roy G. Biv energy spectrum. The inner segment is going to have organelles and the nucleus and some of the neurotransmitters that then are going to synapse with the bipolar cell. All right? The cone cell is all about your color vision. All right? The rod cell is going to be attached again with its inner segment, and its inner segment has a lot of these little discs. And what these discs contain is a specialized pigmentation molecule called rhodopsin. And rhodopsin is going to be a molecule that we worry about because it's going to be what is sensitive to the energy levels that gives us our nighttime vision. It's also sometimes known as visual purple. And rhodopsin is going to be the retinol, so the reason why you want to eat carrots and get your vitamins, because what rhodopsin is made up of is those different molecules, okay, keratin and retinol, okay. So the visual pigments are going to be where light is absorbed, and light is then going to be utilized to make changes to the rod or the cone cell. For the rod cell, they have a chemical known as visual purple, which is rhodopsin. It's made of opsin plus retinol. And retinol, again, is synthesized from vitamin A. So this is why you have to get the fat-soluble vitamin A in your system. Okay. For the cones, they are sensitive to the spectrum of light that's going to have color associated with it. And the theory is that, and it's a theory, remember, so the theory is you have cone cells that are going to be sensitive to red, you can have cone cells sensitive to green, or you can have cone cells sensitive to blue. So if we look at it, a blue cone cell is sensitive to the wavelength of about 425, and its best sensitivity is somewhere around 450, 460, and then it can all the way be sensitive somewhat to around 500 to 540. The green wave, the green cone, is going to have its wavelength from around 460. Its best sensitivity is around the 550 50 range for nanometers, and then it can go all the way up to the orange spectrum, so in the 625. And then the red one starts right around 480 and goes up to 700. And so what happens is as light comes into our eyes, it's going to be traveling at different wavelengths. White light is a combination of all the wavelengths. Black light is an absence of those wavelengths. So if the image we're looking at is sending us a bunch of red, and therefore we think it's red because it's sending us energy that's in the spectrum of 600, 650, 700, that's going to trigger the red cones, maybe depending upon the wavelength, a little bit of the green, but not the blue. All right? And if something's coming in at purple, it's going to be triggering more of the blue by itself and not the red or the green in there. Okay? So we have three populations of cone cells. Now, for some people, they are blind, colorblind. And so what ends up happening is somehow they have a dysfunction in their cone cells. It might be just the red, it might be just the green, it might be both red and green, but there's a dysfunction in the cone cells and their ability to respond to certain wavelengths, and therefore that person has an inability to see in certain colors. so they have to use mostly gray, which is the rod cells. Okay. So the rods and the cones are your photoreceptors, and what happens is they are sensitive to photons, which are energy molecules in light. And what the photons do is they travel and are going to interact with the rhodopsin, or the visual purple protein molecule in the rod or cone cell. And then what they do is they change the rhodopsin from a cis or a trans formation, so that's chemistry terms, and they activate the opsin component. Right? And so when they activate the opsin component and they change from 11 cis form to 11 trans form, that is going to change what goes on in the rod cell in terms of potassium levels, and by changing the potassium levels, we then now have the ability to potentially release neurotransmitter and activate the synapse between the rod and the bipolar cell, or the cone and the bipolar cell. Okay? Now, we know that specifically with rod cells, there is something called bleaching that can occur. So it takes time for the rhodopsin molecule to return to a state that it's ready to be activated again. And so that is what happens when you walk into after a movie and you're in the middle of the day and it was dark and you have all your rods and cones sensitive to the low light energy, and then you walk outside and all that bright light kind of makes you feel like you've bleached out and you can't see for a few seconds. Or you're at night, you're in the dark, and then someone turns on a light or flashes a flashlight at you, and then you lose your ability to see at night. So that bleaching effect would be when you have a bright light stimulus that kind of simultaneously causes all of your rhodopsin to undergo a change, so you get what your brain sees as a brightness, and then for a period of time while you have to wait for those rod cells to reset, you have an inability to see at night. All right? You can also have a night blindness not due to whiteout or bleaching, but because you have a deficiency in vitamin A, and therefore you don't have as much rhodopsin available in your rod cells. All right? So how does this work? Typically, you're going to have, if we start at the beginning, light is absorbed, and when the light is absorbed, the activation of the opsin occurs, and it becomes 11 trans retinol. All right? And then through that 11 trans retinol, it's going to change sodium permeability, and that sodium permeability is going to end up causing, and I said potassium earlier, and I'm sorry, but it's sodium that's going to interact and allow a synapse and a release of a neurotransmitter to interact on the bipolar cell. Graded potentials happen. The bipolar cell then gets a stimulation, typically because it's going to have temporal and spatial summation of multiple things it's talking to, and it's going to generate an action potential that then, with the release of neurotransmitter, can get the ganglionic cell to move. Right? And so that's going to the brain. Now, what's going on in the rod cell while this is all happening is, in order to get this rod component, visual purple, back to an ability to be responsive to light again, the 11 trans retinol needs to become 11 cis retinol and reform with opsin. And so that process takes time. Right? And this is why you can bleach out your nighttime vision very quickly with a light being shined in your eyes or some type of whiteout process, and it takes time to be able to see again with your rod cells. Right? You also see this with walking in snow. With all that reflective energy on the snow, the white, it can make you have a hard time seeing because you're basically bleaching all your rod cells simultaneously. Okay? So with light and dark adaptation, at dark or at low light times, we tend to utilize our rod cells for our vision, and they are the most receptive. When we have good light and good ability to see in daytime, we're going to use our cone cells because the light is actually going to bleach out our rods and therefore make them less effective. That's part of the reason why your periphery is not always that great, because there's more rods in the periphery than there's cones. Right? For the visual pathways, so again, we have the photoreceptors, the change in the chemicals that are in the cones and the rod cells. That leads to changes in membrane potentials, That leads to a neurotransmitter talking to the bipolar cell. The bipolar cell talks to the ganglionic cell. The ganglionic cells travels on the optic nerve, cranial nerve 2. That cranial nerve is taking information towards the occipital lobe. Now, for the eye, you're going to actually have the 2 synapses in the eyeball itself before you get to the brain, and one's between the photoreceptor and the bipolar cell, and the other one's between the bipolar cell and the ganglionic cell. All right? Then once you go into that ganglionic cell, you're now proceeding towards the optic nerve, and the optic nerve, you have 2 of them, is going to reach the optic chiasm. And at the optic chiasm, some of the nerve fibers are actually going to cross over. So we have 2 synapses in the retina, and then the optic nerve is carrying information. Now, some of this information from the right eye is going to stay and actually go to the right occipital lobe, so it's going to stay ipsilateral. But some of the information is going to cross and go contralateral and actually go to the left. So what they're trying to show you here is the yellow region is the left eye, which is the medial part, which is getting information over here, is actually going to synapse with its rods and cones, be carried on the optic nerve of the left eyeball to the optic chiasm. It's going to cross at the optic chiasm and continue on to the certain nuclei in the thalamus, and then continue to the occipital lobe, and specifically in the occipital lobe on the right hemisphere. And so this is why your right brain can see part of what your left eye is doing, right? Versus this over here, again, the red is coming this way, all right? So we have crossover, but then it doesn't really show here, but there are certain regions over here of red that actually are going to stay, and you can kind of follow this black line here that stays on the right side. So some of the takeaway is not all visual information crosses over. Some is going to cross over and some is going to stay on the same side. And all of that crossing over occurs at the optic chiasm. Okay. The ear. The ear has 2 of our senses. It's got our vestibular cochlear system. So it's going to have vestibular, which is the equilibrium, the dynamic and static equilibrium, and it's going to have the cochlear component, which is the auditory. Your ear is divided into, from the tympanic membrane out, that's the external ear. The tympanic membrane to the little oval and round window of the vestibular cochlear, little snail-looking thing, that's the middle ear, and that's where we're going to see our ossicles and our air chamber that connects to the nasal pharynx, so the air pressure issues. And then the inner ear is going to be where we have the cranial nerves interacting with hair cells and mechanostimulations to give us information on static and dynamic equilibrium, as well as auditory sounds and frequencies. All right? So the external ear has the auricle, or the external acoustic meatus, and it's an opening for the canal that allows sound waves in the air to be focused and directed towards the tympanic membrane. All right? You have the bones, the external acoustic meatus, which ends at the tympanic membrane, and the tympanic membrane is a thin, transparent sheet that separates the external ear from the middle ear. So if you look at someone's healthy ear, when you look at the tympanic membrane, it's semi-transparent, so it's kind of silvery, but you can kind of see behind it. That's why with little kids, they can kind of sometimes tell there's fluid behind the eardrum. Okay. For the external ear, we have these specialized glands which make wax. They're called sermonious glands, and they secrete sermonium. And part of what that is to help trap any object so it doesn't get into the ear and only sound waves continue to pass. So this is what tries to help us from getting microorganisms growing in our ear. Right? The middle ear is going to be from the tympanic membrane to those windows on the cochlear and the vestibular component. So this is how we have an air space behind a membrane connected to our nose. So when we go to altitude or go into diving or swimming, we feel changes in our ears and changes in our cavities in this middle ear. It's going to close and protect 3 auditory ossicles, so 3 bones, so the malleus, the incus, and the stapes, so the hammer, the anvil, and the stirrup. And those bones are, just like any bones in our body, susceptible to osteoporosis and problems, and could be the reason why we might have hearing problems with age. Okay? The inner ear is going to be divided into the vestibular component, which is static equilibrium, semicircular area, which is going to be where we have the dynamic equilibrium, and the cochlea, which is going to be related to sound waves. So the semicircular canals are oriented in the X, Y, and Z plane, so you can have sensation of moving up and down, left and right, and forward, backwards. And then the auricle and the saccule are going to play a role in the vestibule with your dynamic equilibrium, so that way you know if your head is pointed towards your toes or pointed up towards the sky. Okay, so it's like head location. All right, and you have your round and your oval window in that vestibule, so you initiate the movement of the sound wave from the air to the fluid in the cochlea through the vestibule, and then the cochlea interprets what that movement of fluid is going to end up being sound-wise. Right? The vestibule encloses the saccule and the auricle, and it provides sensations basically for head location and a little bit of linear acceleration. So when you start your car and you kind of feel like your head's being thrown back or thrown forward when you slam on your brakes, that's linear acceleration. The semicircular canals are going to contain fluid in them and are going to have receptors that help give you information on angular acceleration. So whether you're turning, whether you're sliding, you know, relationships in kind of a more three-dimensional range. And the cochlea is going to contain this membranous labyrinth and this bony labyrinth background that is going to allow hair cells to interact and be manipulated and moved based on fluid movement, and therefore give you sensation of sound. Right? There are 2 key pieces on the internal ear that relate to the sound bit. There's the round window and the oval window. Right? Now, oval window plays the first step in the process of getting information from the tympanic membrane and the bones into the cochlea. So the oval window is what the stapes, the bone, is physically attached to. So anytime the stapes is physically manipulated and physically moved, it is going to physically then distort and change the oval window itself. So this is how the bone is physically attached. So these are direct connections. And so if the stapes is being triggered at a certain amplitude and frequency to move, that then is going to translate to the oval window is going to be triggered to move at a certain frequency and amplitude. And that then will cause changes in the connecting fluid behind the oval window. Now, the round window is going to be a more membranous partition that is going to separate the perilymph from air spaces of the middle ear. All right? Your equilibrium is your sensation that gives you information of spatial awareness in some ways, of where my head is in relationship to my feet and where I am in relationship to sky and ground. And there are specialized hair cells that are going to be manipulated with changes in fluid through the semicircular canals that are going to get deformed when your head starts moving in different directions. Okay? So when we look at the semicircular canals, they are continuous with the auricle, and as you can see, there are these little hair cells, these near the ampulla, these little enlargements that are going to be tied into the fluid, and these hair cells are then going to be triggered as changes in the fluid occur and trigger vestibular changes in the vestibular cochlear nerve. Okay? So when we look at it, we're going to have the ampulla, the enlargement. So this is the fluid, the endolith within the ampulla. We have these specialized hair cells that are projecting upwards, and then we have this cupula, which kind of makes the hair cells a little bit better able to sense changes in the endolymph and has it move. We have the cristia down here, and we have these specialized cells embedded, attached to the hair, and then attached to the sensory neurons. And so what ends up happening is as your fluid moves, it's going to make the cupula and therefore the hair cells move as well. And so this is how you sense changes in direction. And I can go through this a little bit more in lab and how you have to have, the indolent has to move at 2 degrees per second. So there is a potential that you are turning or changing direction too slow to trigger a change. And the other aspect of the indolent is if you reach a constant steady state in a turn, you will stop sensing it because these neurons will adapt. So there's 2 ways we can manipulate or cause problems with semicircular ducts. We can either have subthreshold, so 2 degrees per second is threshold. We can have subthreshold turns or subthreshold movements, or we can have adaptation movements where we get in a turn for such a long period of time that we no longer feel we're in a turn. Okay? For static equilibrium, we're looking at the macula, the statoconia, and we look specifically at this area which is known as the otolith. And it's a mixture of gelatin matrix with these calcium carbonate crystals. And so think of this as rocks in your ears. And so when we have this area, we have, again, hair cells tied to this gelatin matrix with rocks. And what happens is if I point my head down, the rocks fall forward. And if the rocks fall forward, the hair gets pulled forward, and then I trigger and I know my head's forward. If I pick my head up, the rocks fall back and the hairs fall back. And so this is, again, another simple little thing that can be basically tell us our head position of pointing down, pointing up. And there's a way we can manipulate it and kind of screw it up with these semicircular canals if we start moving our head around while we're in a turn. And I can talk y'all through that. It's a spiral death, and this is what potentially killed John F. Kennedy in his plane crash. Okay? And so that's the cartoon there. For the hearing component, we got to go into the cochlear region, and we have a bony component, and then we have a membranous component. All right. Now there's 3 chambers. Okay. You have the scala vestibuli, and down here you have the scala tympani. So the more superior is the vestibuli, the inferior is tympani. And then you have your cochlear duct. And then on your cochlear duct you have this little specialized area which is composed of the tectorial membrane and the basilar membrane. And if you look, this is where the neurons are coming in and attaching. So it's within this cochlear duct that you're getting the specific changes to hair cells that are going to trigger sound. All right. So that's kind of the cochlear area is going to be the one that's tied to hair cells. All right. So for hearing, sound waves trigger the tympanic membrane. The tympanic triggers the auditory ossicles to vibrate and change, and they are then going to trigger changes to the round and oval and round windows, and that's going to change liquid movement. All right? And so the perilymph and the endolymph are going to change based upon the frequency and the intensity of sound waves hitting the tympanic membrane and changes to the tympanic membrane, which translates to changes to the auditory ossicles, changes to the oval window, changes then to the round window. Okay? So again, the 3 key spaces are the vestibular cochlea, also known as the vestibular duct, the cochlear duct, the scala tympani, or the tympanic duct. The functional area in the cochlear duct is known as the organ of Coti, and it's going to be where we see the tectorial membrane, the basilar membrane, and the connections to the nerve. So here's the organ of Coti. And the hair cells are going to be triggered here. Then you have your nerve cells tied to those inner hair cells, and you have your tectorial membrane, which can change and move based upon the vibrations of the liquid. Okay? So how do we know sound? Sound waves hit membrane. Membrane changes ossicles. Ossicles trigger changes to the oval window. Changes to the oval window are going to cause waves to start being generated in the perilymph, and that perilymph is going to travel through the scala vestibuli and then through the scala tympani. As they go through the scala tympani, they are going to be released by the oval window. But as they're going through, all right, vibration of the basilar membrane is going to occur, and this vibration is then going to cause the hair cells to interact with the tectorial membrane, and then information is relayed through nerves. All right? So as we wrap up, we hear from a range of 20 hertz to 20,000 hertz. All right, pitch of a sound is based on frequency. Amplitude is going to be intensity. With age, of course, overuse and damage occurs, and so our ability to hear in both range and intensity changes. Hearing damage can occur to the tympanic membrane. It becomes less flexible. It can occur with bone changes, just with any bone, damages to the bones, damage to the cartilage, damage to the interactions between the bones, and the round window may actually ossify, so we may have problems dissipating the perilymph vibrations. Intensity is measured by decibels, and we tend to like things at around less than 70 decibels. Your hair dryer is around 70 decibels, so that's why long-term use of it kind of bothers your ears. Anything at 100 or the lawnmower 120 or 200, jet engines, that's really loud. You should be wearing hearing protection. Okay, and so that is our class for special senses. Please read the book. There's a lot of details I've had to skip in time to try to make this less than an hour. So please, please, please read the book, and then come ready for lab to do eyes, ear models, know your muscles of the eye, your cranial nerves that control the muscles, the 3 layers of the eye, the special features of the eye, and the same thing with the inner ear and the ear. And then we'll talk a little bit about how we screw up our vision and our balance and that leads to motion sickness and spatial awareness issues, which in flying community means bad things in an airplane. Okay, I will see you in class.